I would like to introduce our speakers from the Kids First Data Resource today, Dr. Vincent Ferretti from St. Justine University Hospital and Dr. David Higgins from Children's Hospital of Philadelphia.
Thank you, Valerie. So webinars are always a very good motivation for the team to develop new features or to enhance existing ones. So today I'm very pleased to present you the last version of the data portal that was actually just released 20 minutes ago. So that was quite right on time for the webinar. Our objective for this webinar was to deliver two things, a new studies page and a new variant search tool. This variant search tool is still simple, but will be used as a foundation for more complex searching tool for querying our variant database. So just as a background, maybe I should put this in share mode. So until today, we had two different ways of exploring Kids First data, each of them presenting its own view of the Kids First data set. So the first way, the first tool is the file repository. This is the first tool that we release in this project. And the file repository is used to create a list of genomic files of interest. And so you select the files, like sequencing file, variant call files, any kind of files that we have as a data source for the Kids First data set. And then so you can select them using a very a long list of criteria, clinical filters, or 5 genomic filters, like I want all the sequencing reads, I want all the I don't know, the pathological reports and so on. So you can select those files, and then once you're happy with your set of or your list of files, you can save it and push it to the nice computer platform Cavatica, where you can do more analysis using workflows and so forth. So that's the file repository. So it provides a file view of the data, of the Kids First data. On the right side, you have the cohort builder. This is a quite sophisticated tool to create virtual cohorts of participants. So this is a participant view. You get a list of participants that you can save. As I mentioned at the last webinar, the cohort builder is now integrated with the variant Workbench, which means that once you get your set of participants, you can analyze them in the variant Workbench to explore the variant present in these participants and do a lot of any kind of analysis actually on this compute platform as well, which is the variant Workbench. So we have here two views, the file views and the participant views. But surprisingly, there's one view at least missing is actually the study views. So as a matter of fact, the only place in the portal currently to see a list of Kids First studies is in this tiny bar chart that you can see probably here, which gives the number of participants per study. And you can see this tiny chart is getting crowded because as the number of studies increase in Kids First, well, you add more and more bar in this chart, and basically it's becoming unreadable actually. But the funny part is that this is the only place in the portal that you can see the complete list of Kids First studies. So we want to remediate this situation, and that was our first objective. It was to create a study view of the data for the Kids First portal. And so we create a new page, a studies page that we call, which provide a searchable list of Kids First studies. We also introduced a new study fields for better filtering these studies. For example, we introduced the field study domain, cancer, birth defect. So that's interesting because actually currently, before this release, it was almost impossible to get the list of cancer studies in Kids First or the list of birth defect studies in Kids First. You could do that indirectly by using the cohort builder. So you could find the participant with cancer or the participant with birth defect, and then you look at the studies they belong to. But it doesn't mean that if a participant has a cancer that it belongs to a cancer study per se. So now you can do that with the new tool. So you can actually select or filter the kids first study by domain. We also added a new field called program. So we do have many programs in Kids First. So we have Kids First, Target, and so forth. So you will be able to filter by program. Also, we created a broad category called data category, a broader field called data category. So in the file report, we have a field called data types, and but it was quite very. It's still very long list of detailed different types of files of data. So we wanted a broader category. So we grouped those data types in a smaller number of categories like sequencing reading, SNV, CNV, transcriptal profiling, structural variant, radiology, pathology, and other. We also created a new code for studies that are easier to remember, communicate, and display on chart, for example. So, for example, I give just two examples here, like KF, which is it starts always by the program name. So Kids First -DSD target -AML. So this is a simple way to remember to replace the long name study names like SWIFT. This is very similar to what we did for GDC, for example, or ICGC. The same idea here. And also we link this studies page to the dbGaP website. So for each study, you can go to the dbGaP study page, the corresponding study page, and we'll link that to the cohort builder. So I'm going to show you very quickly what it looks like. How do I do that? Switch. So this is the new studies page. So you can see, I don't know if you see, well, it's a bit small, but you have on the left, as usual, the facet. So you can select, as I said, cancer studies, for example. You can add criteria, you can add more than one criteria. So these are the cancer studies in the Kids First program. And then you get a list of 6 studies. You can have multiple queries if you want. You can create a new queries, then you start from scratch, and then let's say you can select by, I want studies with the family data in it. So I have family data, and then you can search by experimental strategy, data categories, as I said, family data, program, and domain. Okay, and for each study, clear this. So for each study, what do you have? You have the code, the name, the program, the domain, as I said, the dbGaP, the link to dbGaP, and then you have the number of participants in that study, the number of family, and then you have the data types that are available for the studies. You have the sequencing read, SNV, CNV, and so forth, and the number of files. And then you have a link to dbGaP, and then you have a link for each of them to the cohort builder. For example, if I want to see this study, the genomic analysis of congenital diaphragmatic hernia, you click here, and then it opens the cohort builder with this study selected, so that you can see a summary view of the content of the studies. So, for example, the observed phenotypes with the sunburst, demographic, and so on. So that's the summary, the data available, the available data available for this. And then you have also, of course, the list of participants in that study. So this is the link to the. So next, I want to show you. That's for the study list. Now I'm going to talk about the second objective we had is the variant database, the variant search tool that we developed. So just to remember, to recall what we have. So we do have, since 2 webinars ago, we presented our new variant database, germline variant database. At that time, it was a prototype. Then the last webinar, the last one, we released this as a production version of this database for X01 Investigator. So this database contains currently 6 studies, 5,000 participants, around 215 million unique variant, and 26.5 billion of occurrence. So a same variant can occur or can occur in more than one participant. So this is the number of variant participants, let's see, which we call an occurrence. So 26.5 billion of occurrence. So this become it's getting what we call big data, let's say. So with this number, you do some math and you can see that each variant is carried by 133 participants on average, which is 2.4% of the participants. And then this variant database currently is only accessible to the kit source investigator through the variant workbench. So remember that. So the variant workbench is a compute platform to query and analyze the data, the variant data, using notebooks. And we're using the Zeppelin notebooks platform as a notebook platform. You have the Jupyter and Zeppelin. So we picked the Zeppelin first, but we're now working on also implementing the Jupyter notebook platform as well. So you will have the choice. So in summary here, we take the files, the GVCF, all the files that can be found actually in the file browser. We extract the data from them, the variants. We annotate them again, and then we put this in a format called Parquet, so in a variant database. So it's a kind of big data technology. It's a technology database. So for the variant, and all this is on Amazon. And then the user, the X01 or the kit source investigator can launch their own cluster on Amazon to create their notebooks to analyze the data. And again, from this notebook, you can have access to the participant set built from the Cohort builder, and then you can then analyze them within these notebooks. So that's the framework we have for the variant database. Now, the release today try to open the variant database to a broader public. And then this is what we did. We create a new variant search tool, so which is a very simple form for any user to query variant data set directly from the portal. And you can search for a single variant or a list of variants in a given gene. We have different criteria, search criteria. You can search by variant definition in that format. So chromosome, start, the reference allele, and the variant alternate allele. You can search by gene symbols and all their aliases. Ensemble gene and transcript IDs, amino acid change, clean var IDs, and DBSNP IDs. So this is currently what you can search for. And then again, you will find either a single variant or a list of variants within the gene. And then this list is ordered by functional impact. So the highest impact first and the lowest after. And this tool is also linked to the Cohort builder to get the clinical profile of affected participants. So I'm going to show you now the demo quickly. So here is the menu. So you have studies, and now we have this variant things. Then you have a summary of the current release, data release for the variant database. You have here access to the on-off button to access to the notebook platform, but this is restricted only for kit source investigator for now. And then you can search. So you can search again by gene, so TP53, for example. And then you can see TP53. You can search by claim var ID. So let's say 5600, and then you find a variant. And then you have the list of variant found. You can search by, again, RS number. This is the smallest RS number I know, RS10. Find it. I'll go into detail about the result in a minute. And then again, you can search by ensemble ID. So for example, AN, SG, and then you have the list of ensemble ID. So again, so with this search tool, you can find either one variant or a gene. And when it's a gene, you get the list of genes, the list of variants for that gene. And this is what I'm going to do now. So let's say type SBDS. This is a cancer gene. So this is a cancer gene. And then you see that I get a list, 1,136 gene variant. And then you have and again, it's ordered by impact. So the highest impact first, And then, so for each, in each row, you have the variant definition, the type, DBSNP ID, a link to DBSNP, the consequences, and all the genes that overlap that position. So here, for example, you have two genes, SBDS, it applies to only variant, but you have also another gene upstream, which is TYW1. And you have the ClinVar annotation, if any. And then you have the number of studies for which you have participants with that variant, the number of participants, the number of alternate allele, the total number of allele, the frequency, and the number of homozygote. So these numbers are clickable. If I click here, I will get the cohort builder for the studies, and there I would click here, I would get in the cohort builder the 39 participants who have this variant. But I won't do it. I would go instead to the variant page. So if I click here, voila, that's the live demo. Right. Okay. So this is what we call the variant entity page. Okay. So for each variant, you have a similar page. So let's go through it. So you have three tabs: summary, frequencies, and clinical association for now. So this is the definition on your left of the variant, the chromosome, the star, the allele alternate, and the reference allele. And then you have the number of study again, number of participants, the frequency, and then you have the reference genome we use. We have a link to ClinVar for the, if any, and a link to DBSNP. And then below you have the gene consequences. So as I show you, this genes actually at that position, this variant overlap two genes. The first one, TY1W1, which is not very interesting because you can see that it's a VEP and PAC, which is a modifier. You can click for all the transcript for that gene, and you have the annotation for all the transcript. But below you have the gene SBDS, the cancer gene. So this is a high impact. You have some score, and normally here you have only the CAD score, but usually you have the SIF, the polyphen. I think we have 6-7 different score and calls. So you can have again SIF to polyphen and so on. In that case, we have only CAD in that example, and then you have the conservation also score. And then you have the transcript. And again, you can show all the transcript. You have two transcripts with high impact and 3 transcripts with less important impact. So that's for the summary tab. And then for the frequencies, you do have the number, you do have a list of kits for studies where this variant is present. So you have for each studies. Here we have 4 studies, and actually you can see that you have cancer study and birth defect studies. So this variant is affecting participants in a cancer study and in the birth defect studies. You have the number of participants for each of them, and so forth, the same numbers as usual. So if you note here, the number are clickable only for those studies that have 10 or more participants. So that's for privacy reasons. So you cannot click on 6 here, but you can click on 14 here. So when you click on this, actually, you go to the cohort builder, where you have actually the filter for that study, this participant in that study. So you have some summary view of those participants who have this variant actually. And then if I go back, you have also the frequencies in different external cohorts, like TabMed, GNOMAD version 3, 2, and 1, and you do have a link to the corresponding page. So let's see if it works. Yeah, then you have the GNOMAD browser open in that variant actually. The last tab is for clinical association. So we do have ClinVar here, a link to ClinVar, and then we just imported the interpretation that we can find on the ClinVar for this variant. So you have the condition, the inheritance, and the interpretation. This is a pathogenic. And then we have this gene phenotype table. So we do have an orphanet. So this is usually it's by gene. So this gene, for example, in orphanet, has been associated with this kind of condition. There's a bug here. We have to fix this. It's a beta release, by the way. So you have two, it's not splitted properly, but you have two conditions in which this gene is involved in orphanet. You have the same for amin. You have link to amin, and then you have different condition in amin for that gene. You have the same for human phenotype ontology, and then you do have a lot. If I do more, we'll have a long, long list, I think, of phenotypes associated with this gene. And then we have the DDD, development disorder, again, and cosmic. So remember, this is a cosmic gene, cancer gene, so it's actually in cosmic for this kind of condition. So yeah, so that's for the first, this is a beta release. We will keep improving this tool, make it more sophisticated, adding new feature, but that's our first version. And now, if I go back to my presentation, yeah, I just wanted to say that for being able to do that, we had to develop a new data indexing pipeline. Basically, we took the data from the variant database. We use Elasticsearch, which is a software for indexing a big data set. And then, as you have noticed, maybe the search is very quick. We search among 22, 25 billion of occurrence. It's very quick, actually. So Elasticsearch is a very performing system, and we have a big cluster. So we index currently only 4 studies from our variant workbench, and in summary, we have almost 5,000 participants as well, and 207 million variant and 25 billion of occurrence, and only the size of the index is 1.4 terabyte. Now that was my screenshot as a plan B. Now further work. So we will keep. So this is a beta release. We are aware of some bugs. Have you seen? You saw some glitches for the display. So we will fix that in the coming days, and we'll make it more robust. And but we will add also data visualization in the coming weeks into the study stage. In the Cohort builder, we want to add a biospecimen table with biorepository specific query functionality. So as you saw in the Cohort builder, we have the summary view and the table, the participant view. So we want to add another view, another table of a biospecimen that has been requested for a long time. Our variant search, well, we will add genomic filters. So we'll be able to not only search by typing variant ID or gene ID or gene name, but we will have also filter. Let's say I want all the variant in ClinVar. I want all the variant with high impact. So we'll add some facet genomic filters on the left of the table so that you will be able to search for a variant in a given gene, but also restricted or narrowed down to some filters, actually. We will enhance the integration with the Cohort builder, and we'll create a search API also. So currently it's only web, it's a web interface, but we want to give access to the search tool through an API, so programmatic access. And then for the variant workbench, well, as I said, we will add the Jupyter notebook platform. We will support R, and we would like to develop Python-like notebooks. And this is what you should see in the next webinar in 6 months from now. That's it. Thank you.
Thank you, Vincent. I'm going to start transitioning to David, but while I do that, you do have a question about whether the variant workbench will have all Kids First data in it very soon, or I guess what would be the timeline for that?
Maybe Adam can answer that question.
So this is a really high priority for us is to load and index all of the variants into the variant workbench. But we also wanted to go ahead and release some of these features to begin getting feedback from users, even around a more limited integration. So please, really engage, some of these tools and new deployments. As Vincent said, our model for development really is to release beta versions of deployments, so we can get feedback around how different users can further improve or implement additional features around those toolings as we load and ingest all the data. Now, all the data is available on the Kids First portal, but as you can see, and I'll talk a little bit about later, we're beginning to now empower this data and its processed form, for additional queryability and use. But all of the data will be available, and all of it will be as performant as Vincent just displayed. And I just wanted to take a second to really thank Vincent and his team for really executing an amazing vision for queryability of data in ways that are secure and consistent with the required regulatory settings, but still accelerate the discovery process on behalf of children. So awesome job, Vincent and team. I know that you guys pushed very hard to reach today's implementation.
I really want to thanks, I forgot that slide at the end, but I really want to thank my team and Jeremy Constanza, who is our tech lead. Without him, there wouldn't be any portal. And the team, Adrian Paul, Christophe Botek, who is new. He worked a lot on the Elasticsearch indexing. Denis Bourgard is our PM. Evan Gravel and Francis are two full stack developer, and Luca is the UX person that designed the features and the UX of the Kids First portal.
All right. So thanks so much, Vincent. So I'm going to take a few minutes here to go from looking ahead to these great features that just got launched today to maybe if you're a new user looking to just get started with the Kids First data resource, how can I get started and dive in here? And so when you go to the portal, it can be a little intimidating for maybe a first person. We've got lots of bar charts and pie charts and tabs and dashes. And how do I get started? Where's a good place to begin? And so I want to highlight today in my quick little talk here some of our resources that we've got up here at the top right of the dashboard page in our resources tab. And so if you click that resources tab, that gives you a bunch of our resources that the Kids First DRC has put together to help users, both beginning users and more advanced users, get some support while they are using our Kids First platforms and accessing Kids First data. So I'm going to start by, you can imagine if I click this support button and click that link there, what that will do, that will take you to our Kids First DRC Help Center. And so these are support pages that we wrote. And if we scroll down a little bit on this page, we can see some links to the different types of topics that we have here. And so we have kind of a story that you can imagine from a very beginning of a user who's just getting started into using the different tools that we have developed, and then to a more advanced user who might be analyzing data or needs some specific questions that they're looking for answers. So the other really powerful thing in our Help Center is this section right in the middle, the Studies and Access page. And so as Valerie highlighted at the beginning of our webinar today, Kids First has, we funded 40 different studies, and so far we've got 17 of those studies on the portal, and we're continuing to add more as we get more and more data generated. And so the Studies and Access page is a great place to be able to see a good overview of where are all these studies, what is the status of them, and how can I apply for access? So each study has its own card here, and you can imagine if I click this heart defects, then it gives us more information about this particular study. And then what's useful is these two links here in the middle actually link out to other information. So that top link sends you to the study abstract that was written by the PI of the study itself. And then that bottom link with the PHS, that takes you to the dbGaP application page. And so if this is the study that you want to apply for access to, the link is right there, and then you can click the request access button. So trying making it as easy as possible in order to be able to apply for Kids First data. Once you receive Kids First data, so we have standard deliverables across all of our different projects, and so we run the same workflows and same pipelines across all of our data sets, and that allows them to be interoperable with one another and harmonized together, so you can do these cross-disease comparisons that Kids First is part of our mandate. And so these workflows, you can find information about these workflows as well. These are on our GitHub repo, github.com/kidsfirst. But we also have all of them available on Cavatica in the public apps section. So that way you can look at the workflow more carefully. You could bring your own data and harmonize it to Kids First data, and that can further your research as well. So more information on these is on our websites. I also wanted to mention that our partners at the Common Fund Data Ecosystem have also put together a great series of training resources as well. So there's our grouped according to lessons is how they call them. And so it begins with, setting up, a brief introduction, and then how to register. And so it kind of walks you through with great pictures. I can click ahead. So this is one where they're looking at the cohort builder. And so you can see we've got great screenshots and highlighting and things like that to show off the different features of the cohort builder. And so the CFDE folks have put together a great set of resources as well. And that's a great place to look for more training and more FAQs. They also host workshops, which is outstanding. I got to sit in on one in December. Where we did an overview of how the cohort builder works. And so I want to highlight in particular these two coming up in June. So RNA-seq in the cloud, part 1 and part 2. These are actually going to be using the Kids First portal and Cavatica for that analysis. And so if you're curious about how I might do that, how I might get started, I encourage you to sign up with the CFDE for these sessions. I think they're going to be great, and you'll get a really hands-on experience of learning how to do analysis in the portal and in the cloud on Cavatica. Another great place to get support is at our monthly user support office hours series. So that's hosted by me, but I'm joined by a lot of very helpful folks here at the DRC, members of our bioinformatics teams and members of our data science teams. And so we are available to answer all types of questions and topics that might come up as part of your research. So we say anything from applying to data sets to understanding your analysis and everything in between. So we're available to answer all those types of questions. So normally these sessions are hosted on the second Tuesday of every month. And so if you check your calendar, that's actually today is the second Tuesday. And so special for April, we've actually pushed our office hours to tomorrow, Wednesday, April 14th, and that's at 3 o'clock Eastern time. And so if you are really excited about Kids First after our webinar today, we encourage you to come by office hours tomorrow afternoon. I'll be there, and I'm looking forward to speaking with you and asking any questions that you might have. We also have the Kids First forums. This is a really new feature that we are launching to try to keep those great conversations that we have in office hours, but unfortunately they're limited to that 1 hour a month. And so we're trying to expand and how do we kind of facilitate those conversations across, outside of just that time frame. And so the Kids First forum is a new tool that we have just launched and are beginning to grow. And so if you have questions, we encourage you to post them on the forum as a public place where members of the DRC and other researchers in the DRC community can respond and reply. And so lastly, the place I want to point out is the Kids First DRC help desk. And so that's our email inbox, support@kidsfirstdrc.org. I encourage you to reach out to our support inbox, and we'll help you. We'll review them, and I'll bring in the relevant folks here at the DRC to be able to address your requests.